Welcome to our second podcast on the first week of development. The landmark events that we're going to cover in this part 2 are fertilization and then the very beginnings of the process of implantation and all the changes that our single fertilized egg will undergo really toward the end of our first week of development. So as we have ovulation, the egg leaving the ovary and moving into the fallopian or the uterine tube, we're going to have the sperm that are going to meet the ovulated ovum. And the site of fertilization is typically going to be the ampulla or the enlarged, swollen end of the fallopian tube near the ovary. Now, this is the usual site of fertilization where you have successful implantation and pregnancy, because it's going to take about a week for the zygote to travel down the fallopian tube to enter the uterus and to be at the proper developmental stage for implantation to occur. If fertilization occurs too close to the uterus, then the zygote will be lost or implant low in the uterus and you won't have a successful pregnancy. So, as we look at this and we look at the timing of fertilization, sperm typically are not going to survive more than 72 hours, and it's more typical that you have 48 hours from sexual intercourse and ejaculation for the sperm to survive to fertilize the ovum, the oocyte. The oocyte, typically it's not going to survive more than about 24 hours and is usually fertilized within 12 hours of ovulation. So you typically have a very narrow window for fertilization to actually occur. Now, as we look at the male contribution to this, in the ejaculate of the male, this is the spermatozoa as well as the semen, the fluids, you're going to have anywhere from 2 to 600 million spermatozoa and about 2 to 6 mils of semen. I think our video said about 300 million sperm in that time. And when the sperm enter the female reproductive tract, they are going to move in the direction of the ampulla at a rate of about 2 to 3 millimeters per minute. So they're really moving fairly quickly. In those terms, some sperm can get to the ampulla within 5 minutes of ejaculation, whereas some may take as long as 45 minutes of ejaculation. Now, it can take longer than that for others, and we'll discuss part of that and part of the process of capacitation and how it can be latent sometimes as we move through the rest of the chapter. But in this case, sperm are moving somewhat in random movements. It's not random such as a bacterium necessarily. And what we're going to see first is that half of the sperm are going to enter the wrong fallopian tube. Ovulation typically happens in one ovary per month, and usually the next month it's in the opposite ovary. So half the sperm are going to travel down the wrong fallopian tube. Ladies, this is why men never stop to ask for directions. We're just used to being wrong half the time. Okay, that's a joke. You can laugh. However, for the other half that do enter the correct fallopian tube, they are drawn toward the ampulla by process of chemotaxis, this chemical attractant. And so when you look at the random movements, they're not quite as random because they're drawn in this direction. And here's an example. If we have an attractant in the lower left, a bacterium which is purely random movement, no attraction to this particular substance, this is what we're going to see. But a spermatozoa has a more direct path toward this attractant. So in essence, what we're doing is by sending out this chemical signal, we're making this target larger. And as you get closer and closer, we are decreasing the focal point to the direction so the sperm are going to make contact much more readily with the oocyte. So as we look for evidence of this, we first have seen in sea urchin eggs that there's a peptide that was identified and termed Resact, which acts as a potent chemical attractant, and it's going to have a specific and calcium-dependent effect on the sperm. And again, as we mentioned before, that creates a much bigger target because you're following not just the site or the physical location of the egg, but now this chemical trail. We've also seen in rabbits that capacitated sperm are able to follow a thermal gradient because you're going to have an increasing temperature as you move toward the ampulla. Now, mechanistically, especially with Resact, here is a model of what's thought to happen. Here we see a receptor for Resact on the surface of the sperm. When we bind a Resact molecule to its receptor, you cause signal transduction to occur. In this point, this is going to cause calcium to be lost from the cell. Calcium decreases intracellularly, result in the spermatozoa swimming in a straight line. However, in another case, if you have events, you can actually have turns in swimming. Now, this is when you're getting closer and closer and closer. We definitely have a hyperactivated sperm at this point. But we do also have a model here where we see resact, and we have our signal transduction, but instead of a decrease in calcium, when we have an increase in calcium, that is going to turn the sperm. So losses of calcium, straight swimming, increases in calcium are going to cause a turn to happen, so that by the straight and the twists and turns that are made, we have a more direct path from the location in the female reproductive tract to the sperm, to the oocyte itself. Now, as sperm enter the female reproductive tract, recall they can have a certain number of hours that they have to go through to be capacitated. And so what's going to happen is when we see some uncapacitated sperm, they actually have the ability to bind with cells of the fallopian tube, the oviduct cells in the isthmus. This is the area between the uterus and the ampulla, or this enlarged end. And what's going to happen is that as these cells bind to these oviduct cells, it's going to provide some sort of nutritional support. It's going to slow down the capacitation rate and really give the sperm a longer lifetime. So this is going to increase the effective fertilization capacity of this particular event. The ejaculate, remember, the sperm are going to have upwards of 48, possibly 72 hours. This is going to extend that even further, and it's going to stagger the number and the timing of capacitated sperm actually making it to the ampulla. Instead of all the sperm getting there at one time, if the egg's not there, the sperm are going to be there too long and they're going to die. But by having wave after wave after wave of capacitated sperm delayed by these cells, you have a greater likelihood of a capacitated sperm being in the ampulla at the time that the oocyte is released and fertilization occurring. Here we see a scanning electron micrographic view showing a pseudo color of a spermatozoa binding with some of these oviductal cells. You can see many of the cells of the oviduct have these elongated cilia. Some of the oviductal cells don't have the cilia, and those non-ciliated cells are the ones that will bind to the sperm. Now, this is a great quote. When we think of fertilization, I think you can already see just the combination of spermatogenesis, oogenesis, capacitation. How do you get the sperm and the egg in the right place at the right time? Well, this is a quote by Scott Gilbert. It's in a book. He's written many developmental biology books, particularly on experimental developmental biology. And I just love this quote. It says, fertilization is not a moment or an event, but a process of carefully orchestrated and coordinated events, including the contact and fusion of gametes, the fusion of the nuclei, and the activation of development. And if you haven't already started to get a feeling for this, well, hopefully just in that you can appreciate orchestrated, coordinated, the complexity of just that alone. But here's sort of a cool thing as well. It is a process whereby two cells, each at the verge of death, because you've got a very small window of time for these two cells to get together, and they will die. But on the verge of death, unite to create a new organism that will have numerous cell types and organs. It is just the beginning of a series of cell-cell interactions that will characterize animal development. You know, a lot of people ask me as a developmental biologist, well, when does life begin? Well, when you look at this fertilization, life began millions and millions of years ago. Life is a continuum because you cannot take a dead sperm or a dead egg and ever have a living organism. What I'd like to do now is have another movie, another animation of the process of fertilization, and then we will go on and discuss some of the specific details of the process of fertilization.
Fertilization is the epic story of a single sperm facing incredible odds to unite with an egg and form a new human life. It is the story of all of us. During sexual intercourse, about 300 million sperm enter the vagina. Soon afterward, millions of them will either flow out of the vagina or die in its acidic environment. However, many survive because of the protective elements provided in the fluid surrounding them. Next, the sperm must pass through the cervix, an opening into the uterus. Usually, it remains tightly closed, but here the cervix is open for a few days while the woman ovulates. The sperm swim through the cervical mucus, which is thinned to a more watery consistency for easier passage. Once inside the cervix, the sperm continues swimming toward the uterus, though millions will die trying to make it through the mucus. Some sperm remain behind, caught in the folds of the cervix, but they may later continue the journey as a backup to the first group. Inside the uterus, muscular uterine contractions assist the sperm on their journey toward the egg. However, resident cells from the woman's immune system, mistaking the sperm for foreign invaders, destroy thousands more. Next, half the sperm head for the empty fallopian tube, while the other half swim toward the tube containing the unfertilized egg. Now, only a few thousand remain. Inside the fallopian tube, tiny cilia push the egg toward the uterus. To continue, the sperm must surge against this motion to reach the egg. Some sperm get trapped in the cilia and die. During this part of the journey, chemicals in the reproductive tract cause the membranes covering the heads of the sperm to change. As a result, the sperm become hyperactive, swimming harder and faster toward their destination. At long last, the sperm reach the egg. Only a few dozen of the original 300 million sperm remain. The egg is covered with a layer of cells called the corona radiata. The sperm must push through this layer to reach the outer layer of the egg, the zona pellucida. When sperm reach the zona pellucida, they attach to specialized sperm receptors on the surface, which triggers their acrosomes to release digestive enzymes, enabling the sperm to burrow into the layer. Inside the zona pellucida is a narrow, fluid-filled space just outside the egg cell membrane. The first sperm to make contact will fertilize the egg. After a perilous journey and against incredible odds, a single sperm attaches to the egg cell membrane. Within a few minutes, their outer membranes fuse and the egg pulls the sperm inside. This event causes changes in the egg membrane that prevent other sperm from attaching to it. Next, the egg releases chemicals that push other sperm away from the egg and create an impenetrable fertilization membrane. As the reaction spreads outward, the zona pellucida hardens, trapping any sperm unlucky enough to be caught inside. Outside the egg, sperm are no longer able to attach to the zona pellucida. Meanwhile, inside the egg, the tightly packed male genetic material spreads out. A new membrane forms around the genetic material, creating the male pronucleus. Inside, the genetic material reforms into 23 chromosomes. The female genetic material, awakened by the fusion of the sperm with the egg, finishes dividing, resulting in the female pronucleus, which also contains 23 chromosomes. As the male and female pronuclei form, spiderweb-like threads, called microtubules, pull them toward each other. The two sets of chromosomes join together, completing the process of fertilization. At this moment, a unique genetic code arises, instantly determining gender, hair color, eye color, and hundreds of other characteristics. This new single cell, the zygote, is the beginning of a new human being. And now the cilia in the fallopian tube gently sweep the zygote toward the uterus, where he or she will implant in the rich uterine lining, growing and maturing for the next 9 months until ready for birth.
So now, with all of that detail in place, we finally get the oocyte and the sperm together. The sperm actually encounters this cumulus oophorus corona radiata zona pellucida oocyte complex. We'll just call it the cumulus oocyte complex. That's what many of the videos are calling it. So the first step is for the first sperm to pass through the cumulus cells, the corona radiata, and the sperm are going to release hyaluronidase. This is an acrosomal enzyme that's going to help disperse the cell-cell adhesions between these cells so that the sperm can actually access, through its movement of flagellum, access the corona radiata. Now, after passing through the cellular layer, the sperm and membrane receptors on the head of the sperm will bind to proteins and carbohydrates present on the zona pellucida. So the first sperm protein we're going to talk about is SED1. And we're going to find this expressed on the surface of the acrosome. And this is going to have binding capability on the zona pellucida. Now we also have on the head of the sperm, we have a galactosyl transferase. Now, this is going to bind to carbohydrates, and what we're going to see is that this GALT binds to a protein via carbohydrates. The protein is called ZP3 for zona pellucida 3, and it's going to be this N-acetyl glucosamine residue that's present on ZP3. This is a glycoprotein that is going to bind to the sperm galactosyl transferase. So here in our illustration on the right, we see a scanning electron micrographic view of a sperm binding to proteins within the acellular layer of the zona pellucida. And here we can see that we have a GALT, this galactosyl transferase, binding to the carbohydrate ZP3. But then here's the SED1, and it's binding to other receptors. We don't have the specific name of these present in the zona pellucida. Now we have other proteins, ZP1, ZP2, and so we'll talk about these as we talk more about the binding and the penetration of the zona pellucida. So GALT and ZP3, when these come together, that will initiate through signaling, mostly G protein mediated signaling, the acrosomal reaction. And this acrosomal reaction, we're going to see calcium channels open. We're going to see a calcium-dependent and very rapid exocytosis of the enzymes present in the acrosome. So if we think of mast cells, this is going to be like a degranulation event, a very rapid transfer of these enzymes packaged within the acrosome to the outside and impacting upon the zona pellucida. So this illustration shows we have the plasma membrane of the head of the sperm intact. We're looking at a profile, so that's why it looks very spear-like. Here we have the acrosomal membrane, and after fusion of GALT and ZP3, this membrane begins to break apart, and we begin to release the enzymes present within the acrosome. So now we just have the membrane surrounding the nucleus of the head of the sperm that is then going to penetrate through the zona pellucida. So, as we penetrate through the zona pellucida, this fairly thick layer, we're going to have adhesive proteins that are going to be lost as the acrosome is shed and we've got that inner membrane. What we're going to see is that these adhesive proteins are going to be binding to ZP2. This is going to be the adhesive linkage that actually the receptors on the head of the sperm are going to bind to help pull themselves through the zona pellucida, and we're going to find these throughout the zona pellucida. So we penetrate through via the help of ZP2. Now we get underneath the zona pellucida. We're in this, they call it the fluid zone, between the zona pellucida and the plasma membrane. And the first sperm that fuses with the plasma membrane, this will be the one to fertilize the egg. And this appears to be mediated by this receptor on the surface of the sperm that's called izumo. And izumo is a Japanese word that means marriage. So izumo and an integrin on the surface of the egg plasma membrane called CD9. So the union of izumo and CD9 leads to the fusion of the sperm plasma membrane and the oocyte plasma membrane. That's going to allow the nucleus, mitochondria, centrioles, and the flagellum of the sperm to actually enter into the oocyte. And that then initiates the zonal reaction, which prevents any other sperm from penetrating, fusing, and binding to the oocyte membrane. Now, the zonal reaction, the oocyte releases enzymes, and this is going to modify the zona pellucida sperm receptors. And one of these enzymes is an N-acetyl glucosaminidase. So remember, GAL-T that binds to ZP3 via the N-acetyl glucosamine, this is removing that carbohydrate, so GAL-T no longer has anything to bind to, and it prevents a second or additional sperm from binding, causing too many sperm to fertilize the egg, which would not be compatible with life. So here we have a synopsis of the events. We can see steps 1, 2, 3, and 4, all the way to the point that we actually get the fusion of the plasma membranes, the introduction of the nuclear material from the sperm into the oocyte. And once that happens, now we finish that process of meiotic division in the females. We form the female pronucleus. The other half of that genetic material is going to be in the form of a polar body that is actually left outside of the cell. The nuclear material from the sperm is then going to form and become the male pronucleus. And a lot of this is happening much like we would see with mitosis. We have spindle fibers forming. We see the condensing genetic material forming the sister chromatids. And these microtubules then, these spindle fibers, are going to align both the male pronucleus, the female pronucleus. They're going to align together along an equatorial plane. We form a mitotic spindle. And if you took a snapshot of that process, you would not know you were looking at a freshly fertilized egg undergoing the first mitotic or cleavage division versus any other somatic cell. It's going to look exactly the same. So here we see that we've got the sperm trying to penetrate and get in. One is successful in penetrating. So this is the female pronucleus. We are going to go from having the sperm penetrate. Now we have the male pronucleus. The genetic material is going to begin to condense into sister chromatids. We're going to lose the membrane around those pronuclei. They're going to fuse together, and there we have the equatorial plane. We're going to align all of those sister chromatids up. The mitotic spindle then will pull the sister chromatids apart into individual chromosomes. And so this daughter cell of the zygote and this daughter cell of the zygote will have identical genetic material. Each will have 46 chromosomes. We will then result in a 2-cell zygote and the beginnings of development. I don't particularly like the way they've drawn these because it looks like you've got male and female on one side or the other. That's not what they're illustrating here. They're just showing identical genetic material going to both cells. Here, as we look at a zygote, we're looking at an oocyte, and the genetic material is going to be in blue. We're looking at some of the cytoplasmic components in green. Here we can see an oocyte. We've got a polar body up here at the top. Now we've got the male pronucleus. Here we see the female pronucleus and a polar body that's being ejected. A lot of the green are going to be the mitotic spindles. Now we've got them aligned together, forming a single nucleus that we're getting ready to divide. That's actually the flagellum of the sperm that's left over. And here we have a nice metaphase spindle, sister chromatids aligned up along the surface, and we're beginning to separate those as our first cleavage divisions of development. So fertilization, what are the results then? Well, we are going to complete the second meiotic division in the oocyte. We're going to restore the diploid number. We're going to take 23 chromosomes from the oocyte, 23 from the sperm. Now we have a diploid number for a human zygote. We're going to determine that the species is variable because neither the maternal nor the paternal aspect is going to be completely mimicked in this new offspring because of recombination, because of crossing over. You're going to have a brand new individual that the genetic combination has never been seen before on the planet. Certainly, we're going to result in the determination of the sex or the gender of the embryo. And you would think that when you look at the composition of sperm and ejaculate, that the ratio is 50-50. And we always say that there's a 50-50 chance at having a male versus a female. Well, when you look at the percentages of babies that are born, essentially, when you look at 200 babies, 105 will be male, and slightly over 50-50. You have more males than you do females. They're not sure how to account for that slight difference, but it's not close enough for us to really consider anything more than 50-50. But there are other factors that appear to be involved just between the number of male versus the number of female bearing spermatozoa. Lastly, as we saw in the previous illustrations, when we have fertilization occur, that's going to initiate the first cleavage division. That one cell fertilized zygote, the fertilized oocyte, that is very short-lived. In fact, it's already beginning the process of division when the sperm actually fuses. So that will initiate then the continuation of the cleavage divisions and resulting in all of the stages that we're going to be talking about next. Here we can see fertilization, the single-cell zygote, 2-cell, 4-cell, 8. Then we go to multicellular, and then we're in the uterus. And this process of fertilization here in the ampulla toward the end, the movement by the ciliated cells in the oviduct transporting the zygote toward the uterus takes about a week to occur. And in that week's time, we're going to be at the appropriate stage of development so that we have structures in place that allows for implantation in the uterus. So here we are, day 1 to 2 after fertilization. We are in the fallopian tube, and what we have now is we have a structure that consists of multiple cells. We're still going to be surrounded by the zona pellucida because remember, we just basically got one sperm through the zona pellucida, so by and large, it's intact. Now, the cells that are undergoing the division inside, this is one of the reasons why we have cleavage divisions. And remember, this is cell division without increasing in size. It's because we don't have a lot of room to grow here. What we also may see in some of these early zygotes are going to be a polar body. That's going to be the degenerating nuclear material from that last meiotic division. Now, the cells that occur during these cleavage divisions, we refer to them as blastomeres in these early stages of embryonic development. So here we're looking at 2-cell, 4-cell, and 8-cell blastomeres inside the zona pellucida. That's what's represented here in green, illustrating some of these polar bodies. Each one of these cells is referred to as a blastomere. Now, as long as you have a solid ball of blastomeres, we refer to that as the morula, or the morula stage of development. Now, as we look at the cleavage divisions, we're going to see that it's not always equal, or it doesn't always occur along the same plane in every single organism. Now, humans certainly we do, but we can have different planes. In humans, and mammals in particular, the first cleavage division is said to be meridional, meaning we're going to have vertical planes that are going to separate the cells during these divisions. However, the second division can also be rotational. One cell divides meridionally, while the others divide equatorially or horizontally. So look at the image of how this happens. Here we can see that we've got echinoderms, starfish, and amphibians. Well, we all have this meridional cleavage division that's occurring throughout. But here in mammals, we're going to have a cleavage in our meridional plane for 1 and 2. But then we will also have this horizontal or equatorial division, so that we're already not doing exactly the same thing for all of our cells. So we've already started to develop a pattern even as early as a 4-cell stage in mammals. So our blastomeres not only are going to be divided along different planes, but we're also going to see that some of the cells divide at different times. So it's almost unfathomable to believe that even when you just have 4 cells, the cells are already beginning to distinguish themselves one from another. You just started with 1 cell and you've already begun to do different things. Now, one of the things that's going to happen as we get more and more cells, even at 8 cell and beyond, not only are we confined within the space of the zona pellucida, but the cells are going to be drawn and compact within each other. And this is going to be conferred by cell surface adhesion molecules that are called cadherins, specifically E-cadherin. The E stands for epithelial cadherin. These are calcium-dependent cell surface molecules that bind to other cadherin molecules, and this binding of the cadherins together are going to pull the cells closer and closer together into a very compact ball of cells, and that ball of cells being called the morula. Now, morula comes from the Latin for morus, which means a fruit, in this case, the fruit of the mulberry tree. That's where we get morula stage, but it's the solid ball stage. And the solid ball can be as many as 12 to 15 blastomeres, and maybe slightly larger, but as long as you don't have a hollow center, we stick with the term morula. Now, here I want to introduce a term, because when you talk about the ability of cells to become a number of different cell types, like a stem cell, we can have a lot of different meanings, and we're going to have some terms that sound similar but not exactly the same. Look at the single fertilized oocyte. That one cell is going to give rise to everything in the body. That one cell is referred to as being totipotent, totally possible to produce any cell from that one. And as we look at our early blastomeres, 12 to 15 blastomeres, we see all of those are totipotent. And so it's during this early time, if you have separation of some of these blastomeres from others, you can give rise to a completely genetically identical individual. So our identical twins are made as some of these early blastomeres separate from the others, giving rise to identical twins, identical triplets, and so on. So once again, there are blastomeres, and that's what has to separate if you're going to have identical twins or identical siblings. All right, now we're going to look at a little bit later, 4 and 5 days after fertilization. We're still in the fallopian tube, making our way toward the uterus. But now as we get to a much later stage, the morula, there's many more cells than 12 to 15 here. We're still a solid ball. But now we're going to see that the morula cells are going to differentiate into 2 distinct types of cells. Now, these 2 distinct types are no longer going to be totipotent. Each is not going to be able to produce everything, but we say they're going to be pluripotent or pluripotential, capable of producing many different types of cells, but not all of them. So now we have really distinguished our 2 different lineages of our cells that are going to begin. And remember, we're going to have more than 250 different cell types in the human body. So as we look at our blastomeres, and especially as we look at molecules that are working inside these cells, we have a lot of transcription factors. These transcription factors are proteins that are going to lead to the expression of certain genes. And so as we look at our totipotent blastomeres, we can see that we have the transcription factors eomesodermin, CDX2, and OCT4. Those, if you see these 3 expressed in a blastomere, you know that that blastomere is totipotent. However, what we're going to see is that the outer layer of our morula, we're beginning to change our terminology now. We're going to be referring to this as the blastocyst once we start to form a hollow cavity. So the outer portion of the morula, the shell of the morula, these are going to become our trophoblastic cells, or the trophoectoderm. And what we're going to see in the trophoblastic cells is they are going to begin to express eomesodermin, which is again, this trophoblastic factor that they're calling it. And then we're going to see CDX2. Now, CDX2 is going to block OCT4 and this other transcription factor called NANOG. And the ICM that we see here is the inner cell mass. This is going to be that part inside of the morula that is actually going to give rise to the embryo and many of the embryonic membranes. The outer trophoblast is principally going to function in producing the embryonic portion of the placenta. So the inside, the inner cell mass, we refer to that sometimes as the embryoblast. We're going to see those cells produce OCT4, they produce STAT3, and they produce NANOG, again, transcription factors. So eomesodermin, CDX2, those are going to be trophoblastic cells. And if they lose OCT4 expression, that says, okay, we're trophoblast. However, if you continue to produce OCT4 and lose eomesodermin and CDX2, then you're going to be an inner cell mast cell, an embryoblast, and you're going to make STAT3, and you're going to make NANOG, this other transcription factor. So here is our pathway then of our transcription factor. Here is our totipotent morula producing all of these molecules. Well, cells are either going to become trophoblastic, where they produce eomesodermin and CDX2, and if you produce these, they are going to be inhibitory on OCT4, and they're going to be inhibitory on NANOG. However, if cells are producing OCT4, producing NANOG, and especially STAT3, they're going to produce cells of the inner cell mass, which is going to become our embryonic tissue. Now, in this illustration, this is a fluorescent microscopic image. We have hollowed out the center of our morula, and we're going to change the name of this structure from morula to blastula, but this really gives you an appreciation for the cells in green on the outside. These shell-type cells are the trophoblast cells, and the orange cells in the middle are going to be the cells of the inner cell mass. I would go ahead and include, they show these as green as well. If you're on the inside, we'll call those inner cell mass versus the trophoblast on the outside. Now again, we're still in this 4 and 5 days, but portions of the cells of the inner cell mass, some of the cells are going to undergo apoptosis, which is programmed cell death. The cells are also going to be moving and compacting, and we're going to form a cavity inside the morula. And at this point, we now refer to our zygote as a blastula or a blastocyst, with the cavity on the inside being referred to as the blastocyst cavity or the blastocele. Now, part of this is going to be due to water. We can see trophoblastic cells via the sodium-potassium pump. They're going to move sodium into the blastocyst cavity. Water is going to follow, as we learned in the kidneys and how we conserve water. And so this is going to then lead to the process of converting the morula to the blastula, and we will now result in a zygote that looks like this. We still have our blastomeres. We can see our trophoblast on the outside, the blastocyst cavity, but now we've got the inner cell mass or the embryoblast cells that you can see sequestered to one side of our developing embryo. Now, we again are developing a pattern here, and we refer to this part of our zygote as the embryonic pole or the embryo pole. Here is a basically light level microscopic image. Here we have an early blastula that we see. We're starting with a very small blastocyst cavity, not a whole lot of cells that comprise the trophoblast or the inner cell mass, and then a little bit later in development we can see the ever increasing blastocyst cavity, trophoblastic cells on the outside, and the cells of the inner cell mass or the embryoblast on the inside sequestered to the embryonic pole side. Now, at this point, we begin to lose the zona pellucida. The embryoblast inside, the trophoblast, the inner cell mass, they're going to outgrow the zona pellucida and just rupture through and just burst through the inner cell, the zona pellucida. And as we do that, what we're going to see is that, especially cells toward the embryonic pole and the inner cell mass, they are going to protrude and squeeze themselves through a hole in the zona pellucida, and it's been captured quite effectively in a lot of light level microscopic images. These are mostly rabbit morula and blastula stage zygotes, and here we can see a blastula that is protruding and extruding itself out of the zona pellucida. Here we see blastocysts present in the fallopian tubes here. Here we see a blastula stage embryo, very thin trophoblast, the inner cell mass. And this particular image, we have a blastula that has adhered itself to the endometrium of the uterus. This is beginning the process then of implantation. Now, again, all of these events are happening 4 and 5 days. So we're approaching the end of that first week of development. And so initially, when we have a blastula entering into the uterus, it is initially going to make contact with the endometrial lining of the uterus. And this initial contact is referred to as the apposition or the capture. When you think of white blood cells in the circulation, they're first going to come into contact with the endothelial lining, but then they're going to roll first. The certain receptors are going to cause them to roll and slow down. Well, this is almost like that rolling and slowing down, and it's mediated by some of the same receptors and events. Because when we look at the trophoblastic cells, we're going to see that we have this carbohydrate receptor called L-selectin. And that's on the surface of the trophoblast, and it's going to interact with the carbohydrates present on the cells of the endometrium. That association is not really strong, but it's going to create some friction, so it's going to slow down the movement of the blastula and kind of hold it in place a little bit longer, so that then the trophoblastic cells can begin to express integrins on their surface. That then is going to bind to collagen, fibronectin, laminin, these adhesive glycoproteins that are between the cells of the endometrium, and that's going to lead to the adhesion or the really tight binding of the blastula and then the further implantation into the endometrium itself. So here we are at day 6. We've had apposition. Now we've had adhesion of the blastula to the surface. Now we're going to completely embed ourselves within the endometrial layer. It's almost like a tick digging into the skin of an animal. I know that's kind of a weird analogy, but that's exactly what's happening, because really, for all intents and purposes, this zygote is a parasite. It's going to require a host to sustain itself. So after we've adhered, now the trophoblastic cells are going to secrete a lot of enzymes, collagenases, stromolysin, plasminogen activator. So these collagenases, proteases, they are going to create a pathway for the zygote to tunnel its way down inside the endometrial layer. It's going to completely bury itself in the endometrium of the mother. Now, as we go through day 6, the trophoblast is going to differentiate into 2 layers. The cellular component, that we've been calling the trophoblast, we're now going to call that the cytotrophoblast, or the cellular portion of the trophoblast, because as this surrounding shell begins to proliferate, we're going to see that we have growth and we have nuclear division, but we're not going to have cytokinesis. The cells themselves are not going to separate. So what we're going to end up with on the outside is a humongous mass, or a syncytium of cytoplasmic material, and you can't distinguish individual cells. And this is going to surround the cytotrophoblast, the embryo cell mass, the blastocyst cavity, and this outer shell is going to be referred to as the syncytiotrophoblast. Now, this syncytiotrophoblast is going to be producing a lot of enzymes to assist with the adhesion and the implantation within the uterine lining. But we're also going to see that the syncytiotrophoblast is going to play a very critical role in forming much of the embryonic portion of the placenta. So here we have our blastula. We can see some of these receptors on the trophoblastic surface. We see we're going to have these L-selectins binding to carbohydrates on the surface. We're going to have first adhesion. just this initial apposition and contact. Then the more strong adhesion is going to be basically facilitated by integrins and cell surface glycoproteins. Now you can see the development. Here we have the cellular trophoblast, the cytotrophoblast, and then on the outside you see these multinucleated masses. These are all going to fuse together into one big mass surrounding the cytotrophoblast and everything else, and these are the syncytiotrophoblastic nuclei that we see that continues the process of implantation until we're completely underneath the luminal cells. We are in the wall of the endometrium itself. So here we are again, day 4, 5, approaching day 7. We can see in yellow, we've got our cytotrophoblast. We're not seeing the syncytiotrophoblasts in this particular illustration other than just at this point of invasion. We've got our embryoblast or our inner cell mass. Now we're not going to distinguish with other components yet. You notice they've put these in yellow. I hate when they color that yellow. For us, we're just going to call all of this inner cell mass at this point, this blue and this underlying yellow floor that you see here. And here's our blastocele or blastocyst cavity. I also want to point out the uterine glands that are present in the endometrium and the uterine arteries that we see. As this syncytiotrophoblast invades, it's going to invade through the vessels and invade through the glands, so that the nutritive support here, the support of providing oxygen and carry carbon dioxide away, is going to come from the maternal uterine glands and the maternal blood as the syncytiotrophoblast gets larger and larger. Now, we already mentioned that implantation is going to lead to the production of hormones, and in this case, the syncytiotrophoblast is going to be the beginnings in the production of human chorionic gonadotropin. The syncytiotrophoblast being that portion that is going to lead to then the formation of the placenta. So HCG is your typical target for your early over-the-counter pregnancy test that you will purchase. And so it's going to take a few days. Remember, this is day 6, day 7 before we begin to produce it in any appreciable amounts, before you're going to have enough HCG produced and secreted into your urine as your kidneys filter it, so that's detected in a pregnancy test. That's going to take a little bit longer. I believe 10 to 14 days might be the earliest possible that you could detect HCG. For earlier testing, you can look for early pregnancy factor, EPF, and this is more likely going to be a blood test that you will have administered by a physician to determine those early stages of pregnancy. Now, one of the last things to discuss here as we're approaching the end of our first week of development is the hypoblast. We saw that term and that location on one of the previous images. The hypoblast essentially is going to be the floor of the inner cell mass. This is going to be the layer of cells that's immediately adjacent to the blastocyst cavity. A lot of people will refer to this in some terms as being part of the endoderm of the zygote, but I really hate to call it that, and for our purposes, we're just going to stick with hypoblast here. We're not going to call it primary endoderm. We're going to reserve that term for what's going to be happening here in the inner cell mass, and what we're going to use then to build a huge portion of our embryo. So hypoblast is simply the floor of the inner cell mass. Now, to sort of bring this home a little bit, we have a small animation that shows some of these early steps of implantation, mostly the apposition and adhesion process, and then we will continue implantation as we move into chapter 3. So here's our movie. In this movie, we have the morula moving down. You can see that now we've converted that into the hollow cavity blastula. It's moving into the uterus. Here we can see the trophoblast on the outside, the inner cell mass. As that approaches, we have apposition there in the uterus, and typically, and we're going to talk more about this in chapter 3, where implantation usually occurs in the uterus, but we have apposition. Remember, this is through the L-selectins. Then we get adhesion, and then we're going to start to form our syncytiotrophoblasts as we get the invasion. The syncytiotrophoblasts is in green, cytotrophoblast here is in purple, inner cell mass in blue, and the hypoblast, they've already started coloring that in as yellow. There we get the invasion of this early zygote. You can see the ever-expanding syncytiotrophoblasts. Here a little bit different angle as we get larger and larger in the first week.